Okay, well, hi. We are now moving into chapter 14. This is the currently the last chapter that we'll have. So we are now looking at some of the details of how memory and learning occurs inside the brain. So we're going to be looking at the cortex, but also a lot of some of the subcortical structures of the cerebellum and the midbrain, which turn out to also play a pretty important role in learning processes. But before we get into some of the things specific about the brain, I think it should be pretty useful for us to do a fairly quick review of some basic learning theories. Now, if you've had general psych, you should have learned these before, but I know sometimes these things need a little bit of a review. I'm not necessarily going to assume that you also have had the psych of learning class and or the cognitive psych class where you might have had a different chance to review these, not to mention maybe a few other classes like potentially developmental psychology, you could review this stuff too. So let's do that. A couple slides here. First with classical conditioning, aka Pavlovian conditioning. You surely remember Pavlov, right? This is the story of how Pavlov discovered that a neutral stimulus, and oftentimes we talk about him ringing a bell, though in the original research, it was actually the clicking of a metronome. And he demonstrates initially that it's a neutral stimulus because the dogs really don't have any particular response to this sound. And that's the definition of a neutral stimulus, is a stimulus that does not really activate a given response from an organism. But he pairs this with a reflex that the dogs already exhibit. We know that salivation is a reflex. And we're going to use the term conditioning here as synonymous with learning. It's really technically synonymous with the concept of conditional learning, right? Learning based on a conditional relationship between, in this case, two stimuli. So we'll say that the unconditioned stimulus and unconditioned response is a stimulus-response relationship that does not have to be learned or acquired through any conditional relationship because it's built in. It's a reflex, right? So in this case, for the dogs, salivating to food was the unconditioned reflex. Food is the stimulus, unconditioned stimulus. Salivating to the food is the unconditioned response. In the case here where we have the metronome clicking, that was a neutral stimulus at the start, but after Pavlov spent several trials pairing the clicking of the metronome as a neutral stimulus with the unconditioned stimulus of the food, then after a certain number of trials, the dogs began to salivate to the sound alone. So now the sound is definitely no longer a neutral stimulus. It is now a conditioned stimulus, and salivating to that sound is now a conditioned response. So we have now modified this behavior, this dog's behavior. It salivates to a different stimulus than it had originally salivated to prior. What we might also call a different kind of learning is called instrumental learning. And one of the core differences between Pavlovian conditioning and instrumental conditioning is that Pavlov's approach is considered to be what we might call passive, right? A stimulus is applied to the animal and the animal responds to the stimulus. Without a stimulus, the claim is that the animal would do nothing. The animal is essentially sitting there doing nothing at all until it receives some stimulus, and then its behavior is purely deterministic in response to that stimulus. But in instrumental theories, animals are acting on their environment, and those actions or behaviors are now shaped by their consequences. And we have two different versions of that. We have the very earliest of this kind of work, and this comes from Thorndike, who gave us the law of effect because he was studying hungry cats escaping his puzzle boxes, showing that cats who successfully escaped the box to get food were essentially being rewarded for escaping the box. And when placed back into the box, they would be more likely to repeat the actions that led them to get out in the first place. So again, the cat does something first. It has to do something in the box to perform a particular response, and then it gets rewarded for that response, and then it's likely to repeat that response again in the future. For Skinner, it's basically the exact same thing, except that Skinner's, as a behaviorist, didn't really like to talk about the idea of a reward, right? Rewards are things that we would describe as something the animal wants or needs, and it has some awareness of trying to achieve this desire or this goal. Skinner, as a behaviorist, was not interested in trying to describe things like needs and wants, but rather he just would merely operationally define the effect of any stimulus. If an animal performed an action, and that action was followed by a consequence, and that consequence happens to increase the likelihood of that behavior occurring again, then we have a reinforcement situation. On the other hand, if that particular consequence happens to decrease the likelihood of that behavior occurring again in the future, then we have a punishment situation. So Skinner's not trying to explain why a given consequence is reinforcing or punishing, merely just operationally by determining in experiment, if that's the effect that this particular consequence has, then we know it's a reinforcer, or we might discover that it's a punisher conversely, right? Hence, of course, the important difference between the term reward and reinforcement. They are not synonyms, right? It's a core psychology thing that you should know, is reward and reinforcement are not synonyms of each other. Reward means to satisfy a need or a desire. Reinforcement merely means to strengthen. It means nothing more than that. And the behaviorist a scheme here is just to strengthen a behavior. Here we have a diagram that shows exactly what we're talking about here. So again, for Pavlov, we have that clicking of that metronome, right? So as the pendulum just swings back and forth and it makes this ticking sound, it becomes a conditioned stimulus after some period of time, because now the dog is salivating to just the sound by itself without being paired with the food after a number of trials. On the case of say reinforcement and punishment here, we might be trying to train a rat in a maze, right? So in one situation, a rat may pick a corridor. If it's faced with a junction and it can either go left or right. If it goes left, it might find some food. And in that case, we might find that the rat has an increased tendency to choose that left-hand corridor on future trials. Even if there's no food present, it has this now new tendency to prefer and choose the left over the right. So in the initial trials, the idea is that the rat's choice of left versus right may turn out to be kind of 50-50. It's like trial and error. It's like a coin toss because the rat has not been trained or received any particular consequence. But if the rat happens to receive a consequence of discovering food on the left-hand corridor, then we're going to see a change in that rat's behavior over time, where now its choice to go left or right is no longer 50-50. It's going to have some preponderance of favoring the left side of this particular junction. Like I said, even when there is not food present, it will still prefer this to turn left here, right? Or conversely, if it goes down that corridor and encounters an electric shock, then we will find that it now has a preference to go the other direction, right? So it is now less likely to repeat that particular corridor in the future. So clearly learning occurs when we see this change in behavior over time. That's exactly the classic behaviorist definition of learning, is a change in performance over time. In Pavlov's experiments, the dogs initially do not salivate when they hear the clicking of the metronome, but after the study is done, they salivate just by hearing the metronome. Likewise, at the beginning of this experiment with the rat in the maze, they have no preference over the corridors, right? It's 50-50 guess, but reinforcement and or punishment can shape their behavior so that they may prefer one pathway over another, right? So we've seen a change in their behavior. But now the next question from a neuroscience side of things, it's one thing, of course, to understand how these stimuli affect the rat's behavior or the dog's behavior, but we want to also know what actually happens in the brain, something must obviously be changing in the brain. And that's that first hypothesis we have right here. There has to be some physical change in the brain to underlie this change in performance. So when learning occurs, or even memory, we're going to talk about both of these here, there must be some kind of physical change in the brain. But the question, of course, is what's the change? And because what we're seeing here is that, especially with Pavlov, this kind of learning could be described as forming an association between the neutral stimulus or conditioned stimulus and the unconditioned stimulus, right? And because those two stimuli become associated with each other, they both now trigger the same response. So we might imagine that associations as a theory of learning could not just be about two stimuli, but also it could be somehow connection and communication between two different locations in the nervous system, right? You could have some activation of the nervous system associated with the stimulus of food, and now you have another connection associated with the stimulus of the ticking of the metronome, and somehow those two regions become somehow connected to each other in the brain, and they previously were not really connected to each other prior to the experiment. But now that they are connected to each other, we have this interrelationship that forms that now they trigger the same response. This change in the brain that underlies learning and memory has historically been called an engram. And we briefly talked about the engram way back in the previous chapter on research methods in neuroscience, because we talked about Lashley's career here, where he spent a large chunk of his time searching for the engram. And he would, he was training rats in terms of these little conditioning experiments. And then he would cut some section of the rat's cortex to see if that would somehow eliminate the conditioned response that the rat had acquired, right? So clearly the idea is that the rat learns something, it develops a conditioned response. And then if there's some connection formed between two regions of the brain, and I make a cut in that, right in that spot where that connection is formed, I should eliminate the conditioning and the association that has formed, right? And we would then see that the conditioned response will now disappear from this animal, right? But Lashley failed, right? He went through all of these different variations of the experiment, and I have here on the next slide a picture that we've also seen before from earlier notes, again, of this map of all of the many different cuts that he made in different animals here, trying to find where do I have to perform the cut in order to eliminate the conditioned response. And after all of these various variations here, he essentially gave up, right? So come back here to the previous slide. His conclusion was that maybe there is no such thing as the engram. And now he has these two new ideas here. And I'm noting here that they are erroneous, but only somewhat erroneous. So we know, if we learned previously, that one of Lashley's key errors was that, as it turns out, the appropriate cut that needs to be made is in the midbrain of the rat, and he was focusing on the cortex. So if he would have been able to get down and to make a much more precise lesion in the rat's midbrain, he could have actually succeeded in his study of undoing the conditioned response. And so there is somewhat of an error in his conclusions here, but I'm saying somewhat because, as it turns out, we might learn that there's a grain of truth anyway to his conclusion. So the first is this idea of equipotentiality. His claim here is that since no single cut damages and eliminates the conditioned response, he believed that the entire cortex worked together as a whole. That's the second part of mass action, and that all of these different cortical regions contribute to the conditioned response. Like I said, there's a grain of truth to this, because as we're going to see, especially as we go through here, we know that learning and memory involves some aspects of the temporal lobe, it involves some aspects of the frontal lobe, and those two parts of the brain are especially important, but potentially even the occipital and parietal lobes play some role, especially because those two lobes process a lot of sensory input to begin with. And if learning and memory rely, of course, on processing visual and somatosensory input, then clearly the occipital and parietal lobes are going to play some role in the formation of learning and memory. So of course, yes, the entirety of the brain is pretty important at all times for all kinds of tasks, right? We don't want to fall victim to the old myth that sometimes is bandied about, the idea that we only use 10% of our brain at any given time. We are more or less using 100% of our brain all the time, right? Though some parts of the brain are a little more important for certain tasks, every part of the brain essentially is active for some reason or another pretty much all the time, right? So we see that Glashley had some problems. And of course, here we get the more detailed answer. So it turns out if we get down into the midbrain, brainstem, and cerebellum, we find that we can affect something called the lateral interpositus nucleus of the cerebellum. How do we suppress it? We're not going to damage it here because we don't want to just totally destroy it. What these researchers have found is that there are certain drugs that can be injected directly into this part of the brain, and these drugs will temporarily suppress the neurons in these regions, right? So if we temporarily suppress the functioning of the lateral interpositus nucleus, what we'll find is that if we try to train these animals to learn a new conditioned response, so we're going to put them through, this is usually done with rabbits, where we're going to try to condition them with an eye blink response. So what you might do, for example, is you can play a tone, and that's a neutral stimulus. But if you pair the tone with a puff of air into the rabbit's eye, that causes the rabbit to blink. Eventually, when you pair the tone with the puff of air, the rabbit will blink just to the tone, right? So now let's suppress the lateral interpositus nucleus and try to train these rabbits to blink to a tone, right? By pairing the tone with the puff of air. What we'll find is that no matter how much we do this, these rabbits will not acquire this conditioned response. We also see that if you have a rabbit that's already been trained to blink to this tone, and you suppress this part of the brain, they will stop showing the conditioned response until that suppression wears off, right? But when let's go back to the first case though, where we're going to take new animals that have not yet learned to do this, and we try to repeat this, pairing over and over again, and they're not showing a conditioned response. And then even after we do that several times, and then we wait for the suppression to wear off, they don't show any sign of having learned, right? They still don't blink to that tone. So our conclusion here is that the function of this part of the brain seems to be involved with the actual acquisition of the response. So we're going to call that the associative learning stage itself, the stage at which the learning has occurred, right, and has been acquired. But let's move into the midbrain now. So in the midbrain, there's another clustering of neurons called the red nucleus. And if we suppress this, what we'll find is that animals are not showing any signs of having acquired the conditioned response while the red nucleus is suppressed. But if we repeat these trials over and over again, even though the animal doesn't show a conditioned response, if we wait and let the suppression wear off, and we present the tone, suddenly the rabbits will start blinking, right? So it appears that the red nucleus suppressing that does not prevent them from acquiring the conditioned response. It simply prevents them from demonstrating the conditioned response. So it seems to play much more of a role in the actual kind of execution of the action itself, right? We see that again, just like in the lateral interpositus nucleus, already trained animals also cannot exhibit a conditioned response while the red nucleus is suppressed. So again, we see there's some issues here where that's an important part of the brain for controlling and activating conditioned responses. Also worth kind of making a little side note here that areas in and around the red nucleus are also affected by Parkinson's disease. And so when we see advanced Parkinson's disease and people have difficulty initiating movements, it can sometimes be because this part of the midbrain is now affected by the disease. So here we're talking about learning in the very simple cases that were very popular in the early 20th century amongst behaviorists. But as we move into the mid to late 20th century, we also are interested in other kinds of changes in behavior that are a result of something beyond simple learning cases like that, simple associative learning cases. Memory, for example. When we learn, we can use this word learning outside of the context of a Skinner or Pavlov type experiment to talk about, say, acquiring knowledge, right? You take a class and you're learning, right? But that kind of learning seems very different because you're storing all this information that you are acquiring, right? And so it seems to be more what we might call memory instead of learning. But fair question is, what's the difference really between memory and learning? Both of them involve change and both in the brain as well as change in behavior. In the case of acquiring information, such as when taking a class, the behavior is not something simplistic like salivating to something or blinking to a stimulus, but rather it's the ability to answer questions, perhaps, right? So it's about a topic, right? So in that case, it's more of a form of declarative knowledge, right? So we're going to revisit Hebb again, who we also talked about previously in chapters like the neurodevelopment chapter. And so Hebb is the one who was one of the really originators of concepts like short-term memory and long-term memory. So he's trying to understand learning and memory in neurons and in brains. He's studying various animals and humans as well. So he does make this distinction that there seems to be functionally two kinds of memories, short-term and long-term. We don't yet have sensory memory, which is another idea that came about in the 1960s, but we're not going to really worry about that. Short-term and long-term memory, though, these are something that might be, if they're really functionally different kinds of memory systems, that means that they might also have very different functional networks in the brain. And so we're going to try to figure that out, right? So first thing we want to do, though, is that when it comes to trying to classify different kinds of memory, maybe we should go a little bit further than just the simple short-term versus long-term. So, for example, sometimes there's some discussion of different kinds of memory systems. And another distinction that could be made in memory is to talk about implicit versus explicit memory. And implicit and explicit memories could take the form of either short or long-term, right? So I have short-term and long-term listed at the bottom. But there's also this idea that sometimes there are memories of things that we can specifically recall, and that's called an explicit memory. So if I can explicitly recall what I had for breakfast today, then that's explicit memory, right? If I can explicitly recall when I learned how to drive, that's another form of explicit memory, right? But implicit memory are memories that clearly we must have, but we can't necessarily describe them. So for example, when learning how to drive, or maybe more accurately here, learning how to ride a bicycle, we don't really have the ability to describe the knowledge that we have acquired. Like when you're riding a bike, what happens is that you get on the bicycle and somebody helps you stay upright, and while you start pedaling and steering, and then they let go and you fall over and crash. And then you get back up and try it again and fall over and crash. And then you repeat this over and over and over again. And after some number of times of doing this, suddenly you don't fall over and crash anymore. Something just kind of clicks, right? And now you have it and you're able to stay upright. And so what actually clicked, right? What actually was that knowledge that just seemed to somehow gel and come together so that you don't crash anymore? And that's the kind of thing we really can't verbalize, right? We don't know really what it is. I mean, we kind of know from a physical from a physics standpoint, but not from an individual standpoint. Individual 5-year-old children learning to ride a bicycle don't know what they have learned, but they have learned it anyway, right? What have they learned? They've learned how to harness the gyroscopic dynamics of a bicycle, taking advantage of a concept called precession, right? And this idea that the forward angular momentum of the wheels as you are pedaling forward, combined with the appropriate adjustments of the handlebars, provides gyroscopic stability. But of course, a 5-year-old can't describe to you the physics of gyroscopes, and yet they still understand them sufficiently to harness that to ride a bike. So that's a form of implicit memory. There's some kind of dynamical skill that's acquired here that is totally disconnected from our explicit ability to describe gyroscopic precession. So clearly that's a different kind of memory, a different kind of knowledge altogether. And so of course, if it's a different kind of memory, then again, with different kinds of memory systems, there may be different memory circuits in the brain underlying implicit versus explicit memories. Likewise, again, for short-term versus long-term memories, right? Just a little note about amnesia here as well. We talk about anterograde amnesia and retrograde amnesia. Retrograde amnesia is your is losing memories from the past. So if you had some brain trauma, if you forget things that occurred prior to the brain trauma, that's considered retrograde amnesia, referring to backwards in time. Anterograde means referring to forward in time. And in this case, if you have certain kinds of brain damage, you might have anterograde amnesia, because in this case, you cannot create any new long-term memories. But amnesia is only a reference to long-term memory here. So I've heard sometimes people say things like anterograde amnesia is also called short-term memory loss. That's not correct, right? That is absolutely not correct. It is just merely the inability to create new long-term memories, right? From working forward in time from after the brain trauma. But as I'm noting on this slide, these kinds of amnesias typically only apply to explicit memories. And of course, that explains why we have certain cliches, such as it's like learning a bicycle, you never forget. Because apparently, yeah, we never really forget that, right? And you can go 40 years without riding a bike, and then you can hop right back on it and ride it again. You might even have a brain injury that causes you to forget the day that you learned how to ride a bike, or to forget that you've ever ridden a bicycle before in your entire life. But nevertheless, you could still most likely get on that bike and ride it. So you've lost the explicit memory of having ridden bicycles, but you still retain the implicit memory of how to ride the bicycle, right? Final review there. So you may again remember from prior classes that short-term memory, the kind of classic idea is that it's a limited capacity system that's only capable of holding 7 plus or minus 2 chunks of information. Now, it may also have a limited duration as well. And in part, that's primarily the result of the fact that if I'm thinking about some piece of information that I want to recall later, it's hard to keep that information active because we are easily distracted by other pieces of information. So over time, our concentration waxes and wanes, and our short-term memory content kind of shifts to something new. For long-term memory, we don't necessarily think that there's any particular capacity limit. Our brains don't seem to run out of space for new memories. And there doesn't appear to be a duration limit either, because no matter how long people live, they still seem to be able to recall events from their childhood. So maybe if humans lived longer than they currently do, it might be possible to, in principle, fill up the brain or find an expiration date for long-term memories, but certainly not within the realm of what we currently understand as the maximum human lifespan. So here we're going to have a theory that in order for memories to be encoded as long-term memories, they first have to come in through short-term memory, all right? So nothing can make it into long-term memory unless it first passes through short-term memory. And there has to be some process that converts short-term memories into long-term memories. So if we're going to have this idea that we're going to have a short-term memory system, and then short-term memories can then be converted into long-term memories, then we have to hypothesize maybe at least 3 different memory circuits here, right? One is a memory circuit for short-term memory. The other is a memory circuit for long-term memory. And the third is a memory circuit to explain how this conversion works. And this is frequently called consolidation. But the idea is whatever you want to call it, there has to be some neurological explanation for how that works as well, right? So how are short-term memories kept in the brain? How are long-term memories kept in the brain? And how are short-term memories turned into long-term memories, right? Also worth pointing out that in modern cognitive psychology, we frequently don't use the term short-term memory, as that term goes back to Hebb in 1949. He also called it temporary memory, by the way, so there's another old term there. But in the 1980s, Alan Baddeley proposed the concept of working memory as maybe an improvement on this idea, because his claim is that, as I said before, short-term memory implies that it has a limited duration, but it's not really the case that it has much of a limited duration. It's much more limited by its capacity. It's 7 plus or minus 2. And so a better, maybe more accurate description of what we use this system for is that we use it when we want to work on information. So information that we have to keep active in our mind, whether I'm trying to remember it and repeat it, or whether I'm just trying to use it to make a decision, it has to occupy these 7 plus or minus 2 holding spots of information or of space in the working memory system. We could further go into a rabbit hole with Baddeley a little bit, but I don't think we really want to do so, because Baddeley also has proposed within this broader heading of working memory that there are subsystems. There's what he calls the phonological loop, which is that inner monologue of hearing your voice when you're trying to repeat things to yourself. And that, separate from that, is called the visual spatial sketch pad, which is your ability to remember images and mental pictures. And then there's also something called the episodic buffer, which is your memory of events. And so now, if we're going to suggest that not only do I need a memory circuit for working memory, Baddeley's theory will propose that we actually need 3 different memory circuits for these 3 different subsystems, right? One is visual, one is episodic, the other is more auditory in nature. But as I said, I don't really think we want to try to go down into these details too much, at least in part because the neuroscience has been a little lacking on that side of the game here for when it comes to understanding working memory. Let's just focus on the general concept of working memory, aka short-term memory, and we can look at a task here, right? What we're going to see from the result of this kind of a study is the importance of the prefrontal cortex, or PFC for short. So it's called a delayed response task. So imagine a simple situation where something like a reaction time task, right? A reaction time task might occur if I have you look at 2 different lights, right? And so you're looking at 2 light bulbs. One is a green light, and the other is an amber light, right? And if the green light comes on, you have to press a button. Yeah. If the amber light comes on, you have to press a different button, right? So maybe there's a little green dot on one button and an amber dot on the other. And when the green light comes on, you press the green button. And when the amber light comes on, you press the amber button, right? So that's a response task. And if we set it up as a reaction time task, your job is to respond as quickly as possible. But we can also eliminate that instruction to respond as quickly as possible by asking you to wait and do something else before you respond. All right, so I'm going to flash the light bulb, and then before you press the button, you have to do a secondary task, right? Maybe you have to write something down. Maybe you have to count backwards by 3s or something of this nature, right? But the idea is that while you're engaging in that secondary task, you have to remember, what was the light bulb that flashed? Was it a green one or was it an amber one? And therefore, what button do I need to push when I'm done with that secondary task? So this now becomes a working memory task, because you have to keep your memory of what color light was active, and therefore what task do I need to perform? And you have to keep that in the back of your mind, so to speak, while you're doing the secondary task. And then when the secondary task is complete, you got to hopefully be able to correctly remember which light was active, and therefore perform your task correctly. If we do some imaging during this task, like say functional imaging, functional MRI or FNIRS, we will find that we see a lot of activity in the PFC here, right? So again, it's suggesting, of course, that because it's a task that has a specific demand on your working memory, that the PFC is expected to be active here. And of course, it is active, right? What we also find is that we can find other converging evidence of the importance of the PFC for working memory, because we know that older adults, as they get older, their working memory performance declines. This is just a normal part of aging. And what we see is that we can do this kind of imaging task with older adults using the delayed response task. And not only do they show a decline in performance, that is, they might forget which light was active and sometimes make mistakes on this task, but activation of their PFC during the task is weakened in comparison to younger subjects who are doing this task. So in that case, this seems to suggest that, yeah, it helps not only help verify that the role of PFC is important for working memory tasks, but also maybe even explains a little bit about what's going on with aging, maybe some decline in the connectivity of neurons in the PFC. Speaking to that point, what we've also have found in some interesting studies over the last decade or so is that people who engage in aerobic exercise, which of course improves blood flow to the brain, it improves the connectivity of neurons in the PFC, right? We see increased synaptic connectivity in the PFC. And even then, as expected, we find that it improves their performance on things like the delayed response task. And something else worth mentioning here, I didn't list it on the slide, but back in that chapter on brain development, there's some discussion of things like neurotrophic factors, brain-derived neurotrophic factors. These are chemicals that stimulate neurogenesis and synaptogenesis in the brain. We see definite evidence that aerobic exercise stimulates the production of these neurotrophic factors, and that maybe helps explain, of course, then why this effect occurs, especially in older adults when their brains are starting to show some decline in neurons and synapses. And if we can kind of re-energize these neurotrophic factors, we can undo some of those age-related changes. Similarly with the hippocampus, right? So a lot of famous studies that we're probably well aware of by now about how important the hippocampus is for encoding new long-term memories, right? So we should probably have learned in general psych the idea of the patient HM who had his hippocampus removed as part of a surgery to treat his epilepsy. Of course, when they removed the hippocampus, they were really trying to remove mainly a portion of his temporal lobe where the seizures were originating, but they also had taken a piece of the hippocampus, which sits directly beneath it. It's really part of the limbic cortex that sits under the cerebral cortex. So they were taking that as well. And so what we found, of course, is that with HM and other people in the years since that happened, who also have damage to their hippocampus, we see anterograde amnesia primarily. Now, in the case of HM, because they had removed his temporal lobe, we found some expected retrograde amnesia as well. But there seems to be that real strong tie-in to anterograde amnesia. So it seems to suggest here that this helps us understand that circuit that I was talking about. There's a short-term memory circuit, working memory circuit that we're going to find in the PFC. There's a long-term memory circuit that we might be finding in the temporal lobe, but sitting in the limbic cortex just under the temporal lobe is that third circuit, the one that's involved in converting short-term memories into long-term memories. That seems to importantly involve the hippocampus. And when we lose that, then we lose the ability to create new long-term memories. As I said, the anterograde amnesia is not short-term memory loss, because even with HM, his working memory was effectively normal. It's just that once he got distracted, because he couldn't create new long-term memories, once his mind got distracted on a different task, he would lose whatever information was previously in his short-term memory effectively forever, right? It could never be consolidated into long-term memories, right? Speaking on the concept of aging here, the hippocampus shrinks by about 1% per year, some point in our middle-aged years, probably in your 50s, right? And what this aerobic exercise research has also found are that, again, following a long-term intervention of aerobic activity in aging adults, they were able to undo this decline, right? So they were able to actually increase the neurogenesis in the hippocampus, and therefore cause it to kind of grow back in a way. So instead of shrinking by 1%, it grew by 1%, right? And so we're actually seeing brain growth here, and that importance of neurogenesis is important here, because we sometimes think that, oh, there's this old myth that we never grow new neurons after we're born. But no, it's really not true. There's still neurogenesis occurring across the lifespan, but one of the primary places for that really is in and around the hippocampus. That probably is, there's a reason for that, which is because this is where memory is forming, right? And maybe we need these new neurons to be able to encode these new memories. And as neurogenesis declines as we get age, this could explain some of the decreasing memory abilities of older adults, except of course, if you're physically active, and that you get some of that neurotrophic factor that can help regrow these neurons, right? Now back to that distinction of implicit and explicit. As I said, amnesia tends to affect the explicit memories, not the implicit. And we saw this, of course, in all of the many studies that were done on HM. So he was really showing a lot of impairment on explicit memory tasks, right? He could not name and describe episodes and events and facts from, say, prior to the surgery, because he had some retrograde amnesia. But of course, if you tried to teach him new facts and information after the surgery, he could not acquire that. So again, this loss of being able to encode new explicit memories. But he still had his implicit memories, right? So, I don't know off the top of my head whether they tested to see if he knew how to ride a bicycle as a child and whether he could still do so. Presumably that would certainly be expected that he could. But there were various clinical neuropsychological tests that they would try with him. One example is something called the mirror drawing task. And in the mirror drawing task, you're looking at a drawing of a star, a 5-pointed star within another 5-pointed star. And your task is to take a pencil and trace a line between those two stars, essentially drawing a 5-pointed star yourself, but just have to kind of color inside the lines there. But you can only see your hand as reflected in a mirror. So when you see that, it's a little tricky, because you expect your hand to go one way, but the feedback is mirror flipped. So you kind of make some mistakes. But the more you practice this, the better you get. And what we find is that HM could practice this mirror drawing test and got better over time. And even though he had no episodic memory of having done the task prior, he still showed the carryover of his experience and practice anyway. So he was acquiring the implicit skill while never having any explicit memory of having done it before. We might even think that it could be worthwhile to think back to the chapter on vision, because remember when we were talking about vision, we talked about the 2-channel theory that distinguished the what channel, which is the visual processing of information for recognizing what things are. And it might be the case that in a way that's an important part of explicit memory formation. And of course, that pathway takes us from the occipital lobe into the temporal lobe, which is where we find a lot of the explicit long-term memories. Conversely, there was the where/how channel. That's more about procedural stuff. How do I interact with this object, and how do I move around it, and so forth. And that was a pathway that takes us from the occipital lobe up into the parietal lobe. And especially in the case of HM, the damage that he had was to the temporal lobe. And so this is why we see this kind of effect on the explicit stuff. But his implicit abilities were intact, right? And so it seems to suggest that, again, different neural circuits, right? One in the temporal lobe for explicit skills and one in the parietal lobe for implicit stuff. So further evidence of the importance of hippocampus stuff here. Let's look at here's another form of memory, yet again, spatial memories, right? Now, for humans, we could talk about spatial memory, but as it turns out, other animals are particularly good with some certain kinds of spatial memory. Rats, for example, right? Rats are really good navigators. And I think we've talked about this before. We've talked about maybe place neurons in rats when we were talking about sleep, right? The idea is that when they're learning place neurons in a food search task, we saw some reverberation of those neurons in their hippocampus while they were later sleeping. And if they were prevented from sleeping, they had difficulty consolidating those memories and were less likely to remember later. This seems to suggest, of course, then that these particular neurons in the hippocampus are performing that very specific kind of task that's related to place learning and spatial memories. And so, of course, another thing to do is to simply just damage, if we lesion the hippocampus in rats, we can see some very specific impairments related to their spatial abilities. Maze learning, for example, tends to be particularly problematic now. Also noting here that there are some bird species that have to remember spatial locations, right? They store food caches. These are things like the Florida scrub jay, for example, stores caches of food and has to be able to go back and find that food again later. And corvids of all kinds actually do this all through North America. These birds tend to have a larger hippocampus than other birds that don't hide and store their food in these little caches. So see, again, suggesting that especially in some of these animals here, the hippocampus also has a separate circuit here important for spatial knowledge and learning here. We can look at a few other clinical findings as well. So I've talked already about Parkinson's disease a little bit and the changes to the midbrain and what happens with damage to the hippocampus and stuff like that. But there's a few other things that we see impact memory ability. For example, Korsakoff syndrome is not especially common. But what happens for people with chronic alcoholism is they can end up with a thiamine deficiency. And of all of the many things that this does to the brain, it can particularly lead to an atrophy of a nucleus in the thalamus called the dorsomedial thalamus. Right? This is a pathway that relays sensory information from the various lobes to the prefrontal cortex. And so what we might find, especially if it's relaying information from the temporal lobe, where a lot of these memories are kept, and if I want to access and recall one of those memories, recollection seems to involve bringing that memory out of the temporal lobe and then bringing it into our working memory, right? Because if I'm going to think about it, it has to get back into my working memory so I can now work with that information and think about that information. So how do I bring information back to working memory once it's been stored and held in long-term memory? So this pathway involving the dorsomedial thalamus appears to be pretty important for that. But of course, with Korsakoff syndrome, this pathway is damaged. So now it is problematic for these people to be able to consistently pull these memories back into their PFC. So what they instead are able to do is they can just remember little fragments of memories, right? And the result of this is that they tend to start making up stories, right? It's like they're trying to develop some sort of a story and reason to explain some of these fragmentary memories they have. And they start doing something called confabulation. Given a few memory fragments, they start just telling wild, elaborate stories about things that never, ever happened to them because they're just trying to fill in the gaps a little bit. Right. And so there's this kind of this like interrupted stream of information coming out of long-term memory, and they compensate for that in the PFC by filling in the gaps. And so that's a particular thing noted with Korsakoff syndrome, is this pattern of confabulation that seems to be somehow connected to that disrupted recall process. Now, we could talk about Alzheimer's disease here too. So what's happening with Alzheimer's disease? We don't know entirely what causes it. There may be genetic factors or some evidence of that. There may be some autoimmune issues going on here where some parts of the immune system are sneaking through the blood-brain barrier and damaging neurons, because maybe some of these leukocytes in the immune system don't recognize neurons and glial cells as part of their body tissue, because they're not normally used to encountering those kinds of cells, right? And if they did, then they could start destroying those cells, right? So anyway, we see the formation of certain kinds of proteins and protein tangles and plaques in the brain that are really damaging to neurons, right? So these are called amyloid beta and tau proteins. A lot of this stuff happens when in early stage Alzheimer's disease. It happens near the hippocampus in an area called the entorhinal cortex, which is a region that surrounds the hippocampus and seems to play a role in taking that knowledge or those memories from the hippocampus and then encoding them somewhere into the temporal lobe. So of course, this explains why the kind of forgetfulness that might look somewhat benign associated with just a decreasing hippocampus that we see in regular aging is one of the first signs of Alzheimer's disease. But of course, it progresses to get worse and worse and worse, because then as these plaques spread, we start to see much more severe anterograde amnesia followed by additional retrograde amnesia. But then also sometimes these plaques might start popping up in other regions of the brain as well, showing other kinds of cognitive impairment as well, right? But again, of course, because it happens a lot in the temporal lobe, we're going to see a lot of the explicit memory systems affected, while the implicit stuff remains relatively unaffected. At the time I'm recording this, we're only a few days from the legendary singer Tony Bennett passing away, who had Alzheimer's disease. And there are some videos that you can find online of him performing in recent years with Lady Gaga. And he's still quite able to remember his songs and perform and sing those songs and the skills that he had as a musician. But his ability to remember a lot of other stuff, more explicit knowledge, was very significantly impaired. 6 or 7 years ago, we lost another musician, Glenn Campbell, who also had Alzheimer's disease, and he had a final farewell tour before his disease got too terribly bad. And he had a similar ability. He could play his songs. He was playing guitar and singing and playing at a high level, but his explicit memories were showing some severe decline. In fact, there are many reports that in his final concerts, he would play one of his songs and play it, flawlessly, and then immediately forget that he had just played it, and he'd play it again, right? And it sounds like a little awkward thing, of course, except for the fact that he was so well loved by his audience and his fans that they were all very patient with him and just cheered him on as he played the same song a second time, again, doing so with great skill, because he still kept his implicit abilities while losing the explicit memories of something that just happened 5 or 10 minutes ago, right? So our summary and wrapping up of all of this stuff about some of these larger brain regions and different lobes of the brain is that, yes, we have many different kinds of memory systems, explicit, implicit, short-term, long-term, and so forth. And that means that there are different memory circuits in the brain. And so, yes, we see then that the two-channel theory of vision with the temporal lobe, parietal lobe distinction is important here. Prefrontal cortex, hippocampus, temporal lobe, also important for that short-term encoding, long-term. Of course, then the thalamus, dorsomedial thalamus, may be important for recall. So with all of these different stages and steps and types of memories, there are different memory circuits. And so we can map out some of this stuff by studying what happens with various forms of brain damage and disease and other kind of situations like that. Now, the second half, I'm going to cut it here, but the second half of these notes, we're going to get down into the cellular level of what actually happens with neurons when we're looking at some kind of neuroplasticity underlying the brain's ability to learn and store memories. But that's going to be for part 2.